Hello everyone, I welcome you back to the topical solutions for your IGCSE Biology. And for today's class, we would see on toward the topical questions for this topic of the movement of the substances, where we would see that how come the diffusion, osmosis, and the active transport, which are the multiple mechanisms by which the substances they move in and out of the cells. So how come the examiner they evaluate your concept of these three mechanisms in terms of the past paper, and we will begin from the year 2011 and we would go till the end of 2025. So here for question number one, they say is that we have got the sweet potato plant and the genus is Pomoya and the species is Patatas, and it has got total of two type of the roots. One are the fibrous roots and the other one are the storage roots with us. What they say is in context of the fibrous roots that they absorb water and ions from the soil, and as far as these storage roots are concerned, so they say that they have got the role to be storing the insoluble carbohydrate, and we know the carbohydrate that gets stored within the roots, this is starch with us. And for the figure 3.1, they are showing you the growth of these roots on a sweet potato plant. So if I were to just take you to the diagram, which has this 3.1 with us, so here if you could see, we have got the sweet potato plant and these are their underground storage organ. And if you were to look here, so these they have indicated that they are the storage roots, and the ones that we see here, so they are the fibrous roots, and these fibrous roots they are absorbing the water and the ions, and the storage roots, they are basically storing the insoluble carbohydrate in them. Now, so for the first A part, they are asking us that we are required to be using the word water potential, and we need to tell our examiner that how come the fibrous roots, which they have shown above, they would be absorbing the water molecule. So you know it very well that the roots they have got these tiny cells in them, and these cells, we say that they are the root hair cells. And the scenario with the root hair cells is that they have got the large vacuole on the inside of them. Now, what would happen here is that we have these sausage-shaped organelles. They are the mitochondria, and they release energy, and that energy is in the form of ATP. And by that, the ions that we have got outside of the root hair cells, they would be moved into the large vacuole with the help of the active transport, and that would lower the water potential in the large vacuole. And once we have lowered the water potential in the large vacuole, then by the help of the osmosis, the water molecules that we have on the outside of the root, they would move from their area of higher water potential to an area of the lower water potential here that we have got here. And that would be that the water molecules are also crossing the partially permeable membrane that we have got. So if you were to write the answer for the total of 3 marks, but you would say that these fibrous roots they have the root hair cells and they have lower water potential in them, like their large vacuole has lower water potential, and that basically happens due to the presence of ions here. And by that, the water would be now absorbed by the osmosis, where the water would move from an area of higher water potential, where this was here in more number, to an area of lower water potential that lies here, and that would happen across a partially permeable membrane, we say. Now, if we were to move further for the B part, where the total marks the examiner has given are 3, and he asks is that the membranes of the root hair cells they contains the proteins for the absorption of ions. What you need to be remembering at this point is that these ions that we have with us, so they are the charged particles. And whenever the particles are charged, so they are not able to cross the cell surface membranes directly, but rather than there has to be the involvement of the proteins. And those proteins we say they are the carrier proteins that actually allows the movement of the ions for these plant cells that we have got. So for 3 marks they have said that you have to describe that how the root hair cells they are adapted for the absorption of the ions. So if I were to make the diagram for the root hair cell, as I have shown you above, that we have their cell wall, and the cell wall goes like this. And immediately below the cell wall, you see that I have got their membrane, and that membrane is the cell membrane, and in the cell membrane, there happens to be the presence of the specialized proteins. And these specialized protein, we say that they are the carrier proteins with us. Now we have to tell our examiner that how this root hair cell that we have got, this is adapted for the movement of the ions. So we have got multiple points. The first point is that this root hair cell, this has greater surface area to the volume ratio that would allow for more of the absorption of the ions. The other thing is that we have got these carrier proteins, and these carrier proteins they facilitate the movement of the ions, or they provide passage for the movement of the ions. And apart from that, we know it very well that these root hair cells they are thin, and when they are thin, so for sure they would reduce the distance over which the ions would pass. And you may have learned that the ions movement is due to the active transport. And for the active transport, there is a need of abundant mitochondria. So this is another adaptation of the root hair cells that would help them to be absorbing more of the ions. For 3 marks, what you were required to be writing here was that these root hair cells they have greater surface area to the volume ratio, and their cell walls are thin. And when their cell walls are thin, so this would reduce the distance for the movement or for the diffusion of the ions, and at the same time, the cell membrane they have got the carrier proteins in them, and that would facilitate the movement of the ions. Now here you would score 3 marks, but if you forget any of the above point, you can remember this alternative valid point that these root hair cells they have abundant mitochondria that would be helpful for the absorption of the ions. If we were to move further to this question number two, that actually appeared in the year 2011 and for your October-November session, and again here they have asked for the total of 3 marks, that how the water is absorbed by the roots, and we know that the water is absorbed by the plant roots with the help of osmosis. And in osmosis, the water we know always moves from an area of higher water potential, where the water molecules are more, and we know they are in the soil, and they would be coming to an area of the lower water potential and that exists within the roots, and in doing so the water would be crossing the cell surface membrane. So here you would score total of 3 marks. If we were to go beyond part A, so here we have got this part B, and they say is that we have got the young plants and they were grown in the pots of sand for the total of 4 weeks. Now what we are doing here is that we have taken the pots, and in the pots we have got the sand with us, and in the sand what we are doing next is that we are growing the young plants in them. Now, among the various pots which the investigator would be setting in an experiment, so some plants they say that they were watered with the distilled water, and the pH of the water was kept at 7, and by the distilled water, what we mean is that there would be no presence of any ions or the salts in them. At the same time, he took many of the other pots, but he watered them with solutions, and that solution it was having a different concentration of the salt in it, and the salt that they have added into the water that was sodium chloride, but the pH again was kept at 7. And for this investigation, they are saying that we have kept the temperature of the plants at 20 degrees centigrade. So the simple idea is that we would have this plant and this is growing in the sand pot. In one of the pots I would be adding just a distilled water, and in other of the pots we would add the water into them, but that water would have an addition of the sodium chloride in it. Now they say is that the growth of the plant was measured after 4 weeks. And now something that we measure during an investigation or in an experiment is a variable, and that variable we refer to this as the dependent variable, we say. If we were to move forward, they say the growth of the plants that is actually shown in figure 4.1 in the form of a graph, where they have presented the percentages of the growth of the plant that were watered with the distilled water. Now, if we were to look on this graph, the important point is that you are required to be looking on toward the y-axis. and this y-axis would be presenting the dependent variable that we were measuring. And here we have got the x-axis where they have shown you the concentration of the sodium chloride. So you see that this is something we have changed in the investigation. So this is the independent variable, and this independent variable is coming on toward the x-axis. So if I were to be describing this graph, what I notice is that when the sodium chloride is kept at zero arbitrary unit, so the growth of the plant is max and that is 100%. The time we keep on increasing the sodium chloride concentration, so you see that the growth of the plant, this is decreasing until at 600 you see where the sodium chloride concentration is the highest. So the growth of the plant is zero with us. Now, the overall trend, if we see that there is a decrease in the steepness of the graph, that is indicating a decrease in the growth. But what I see at the moment is that the max, you could say, steepness of the graph that I see is it's lying here between 300 to 400 arbitrary unit of the sodium chloride concentration which they have given us. So here the growth at 300 to 400 concentration of the sodium chloride, this decrease in the growth is faster. Although the decrease is here and at this point of the graph and at this point of the graph, but that decrease in the growth is not as much steeper as we have got in this part of the graph. And if we were to just note down the values here, so you see that here the growth of the plant, this is nearly 55%. And if I were to just come on toward this point, so you see this is coming somewhere halfway around here. So here the percentage is 25. So if I subtract 25 out of 55, so the answer that I would be getting, that would be 30%. So we see an overall a decrease of 30% at this concentration of the sodium chloride that we have. So here I have told you multiple point in term of description, and this is something the examiner has asked here for the total of 3 marks. They say is that you are required to be describing the results which are shown in figure 4.1, and they say that you would gain the credit for using the figures in the graph to support your answer. So here you must know for your exam that in description, if you are giving your examiner a reference of these values and you don't quote these values, then ultimately you would lose the marks. So if I were to just quickly take you to my answer that I have explained above, as I said that at zero arbitrary unit of the sodium chloride concentration, the growth of the plant is 100%. You see, at zero concentration, the growth is 100%. And if I were to move above, I have written it very clearly that when the concentration of the sodium chloride is increased from 300 to 400 arbitrary unit, so as I have calculated above and told you above, that a decrease in the growth is steepest and it goes from 55% to 25%, an overall decrease of 30% is seen here at this concentration of sodium chloride. And at the last point, as I have said above, that at 600, the growth is 0%. So you would have to write this third point for scoring 3 marks, that at 600 arbitrary unit of sodium chloride concentration, the growth of the plant is 0%. Now, if we were to move for the second part, and here the examiner has given you 4 marks, so this means that we are required to be presenting our examiner with a total of 4 statements. What they have asked here is that we are required to explain the difference in the growth between the plants that were watered with the low concentration and those plants that were watered with the high concentration of the salt solution. So if I were to be presenting you with the help of a diagram, so if I just tell you that here we have got a plant cell, this is the cell wall, here we have their cell membrane, and then at the center we have the large vacuole. Now this says that if on the outside of the plant cell, if we people have got lower concentration of sodium chloride, and on the other way around, if we have got a higher concentration of what? The sodium chloride, then how this would impact the growth of the plant. Now you know it very well that for the growth thing, there has to be an increase in the number of the cells, and that increase in the number of the cells, that basically requires the energy, and the energy we get it from the glucose molecule, and that glucose molecule is synthesized within the chloroplast. So what you have to be thinking here is that if we have got low concentration of the sodium chloride on the outside of the plant cell, so this means as that the water would be entering into the plant cells. And when this would enter into the plant cells, so we know it very well that the water would be available for the chloroplast to be making the glucose by the photosynthetic process. And that glucose would be used in the mitochondria for releasing the energy, and that energy would be utilized to be increasing the number of cells. So this is some way where the growth would be taking place. At the same time, the other thing is that when the large vacuole it gets filled up with the water molecule, so this large vacuole would increase in size, and that exerts the pressure on the cell wall, and that brings about a kind of an elongation growth in case of the plant. So that is why we would see more growth in lower concentration of sodium chloride, but we would see a very less of the growth in high concentration of the sodium chloride because less water would enter here, less water would be available for photosynthesis, less energy would be released by the respiration process, and ultimately less energy would be available for the cell divisions, and at the same time, without the large vacuole getting filled up, at high concentration of the sodium chloride, we cannot bring the elongation growth in these plant cells. So here I have explained the entire mechanism of how come we are required to be answering it for the total of 4 marks. So we would say that when there is presence of higher concentration of the salt, then the water potential in the surrounding soil, this would become less because the salt are more, then the water potential for sure in the soil would decrease. And with the help of that, the plants they would absorb less water by osmosis, and less water would be available for chemical reactions like photosynthesis. And if the photosynthesis is not happening, so they do not make enough glucose to be releasing energy forward, making new cells. And without water, the cells they can also not elongate. So if we just move on toward this question number three, and this came in your May June of 2014, and here they have shown you the water cycle. Here I must mention it very clearly that water cycle is no longer the part of your syllabus, but the question which they have asked you here for the total of 3 marks, and they want you to answer him that how come the water it enters the root of the trees from the soil, and we know it very well that the water it would be absorbed by the plant roots with the help of the phenomenon which is osmosis, and by that the water molecules they would move from an area of higher water potential that lies in the soil to an area of lower water potential that exists within the root, and in doing so the water molecule would be crossing the cell surface membranes. So if I just take you to this question number four, and this came in October, November of 2014, and here they say that in figure 1.1, we show you two cells. One of the cell that we see at this point, this is an animal cell, and we have got this plant cell with us. And they say that these animal and the plant cells, they appear here as we would see them with the help of the light microscope. Now, if we were to go on toward the question, here the marks are 4, and they say that these cells, both the animal and the plant cells, so we kept them in the dilute salt solution. And then they say that we have moved them to distilled water, and we must be aware of this fact that the distilled water does not has any ions in it. Now, the ask is that we are required to explain that what will happen to each of these two cells when they are placed in the distilled water. Now, if I were to just present you with the help of the diagram, you see here I have the animal cell, and then on the other way around, I have got the plant cell. And we must keep in mind that the plant cells, in addition to the cell membrane, they also have got this cell wall in them. And here I'm making their cell membrane, and after placing them in a dilute salt solution, later they move them into the distilled water, and we know in the distilled water there are no ions. So that basically implies us that within the animal cell, the water potential is low, And along with that, in the plant cell, again in their large vacuole, the water level or the water potential that we have got is low comparative to this distilled water. And in the distilled water, the water potential would be higher. So this means that the water would move from an area of higher water potential that we have got here in the distilled water to an area of lower water potential that we have got both in the animal cell and the plant cell. And ultimately, they would start to fill up with the water. But the difference that we will see here is that when the water would continue to enter into the animal cell, and since the animal cell they do not have the cell wall in them, so for sure they would burst. And if I were to talk about the plant cell, the water that is entering into the plant cell, it would increase the size of its large vacuole. The large vacuole would start to be exerting the pressure on the cell wall. The cell wall that we have here is rigid, so ultimately the plant cell that we have got over here, this would not burst, but the animal cell would burst. So if I just tell you in terms of words that what you were required to be writing here is that the water would be absorbed both by the plant cell and the animal cell, and that would be due to the process of osmosis, and by that the water would move from an area of higher water potential that was there in the surrounding distilled water to the lower water potential that was existing both in the animal and the plant cell, and here the water would cross the cell membrane. But here we are required to be adding this that without the cell wall, the animal cell would burst, but the plant cell, this would not burst, but rather than it would become turgid. Now, if we were to move further, and for this question number 5 that came in May, June of 2015, and they say that we have got a biologist and he made a slide of some epidermal cells from the scale leaf of an onion bulb. So we have got the onion bulb, and from that, the scientist is basically obtaining a scale leaf. And then out of the scale leaf, he is showing you the diagram of the epidermal cell. And that drawing of the epidermal cell that was made by the biologist, if I were to zoom. So here you see that here A part which he is indicating as here, so this is the cell wall with me. The B part that we have got, this is the cytoplasm with us. And the C part that he has given you here, you must not confuse it with the large vacuole. The reason being is that within the nucleus we have got this denser protoplasmic mass, which is nucleolus. So you have to perceive this C structure as a nucleus, not as a large vacuole. And if we were to talk about D, so you see we have got a lot of blackish kind of an appearance of these points which he has given over here, and he is indicating with the help of D. So you just have to assume that these are the spherical units. So for sure they would be the ribosomes that makes the protein. So if we were to just come on toward the question in the part A, they say that table 4.1, which we are giving you at the bottom, it shows the functions of the structures that lie within a plant cell. And they want us to be completing the table where we not only have to give the name of the structure, but also the letter that is indicating it, and we have to be relating them to the functions which they have given us in the first heading. So here they ask us that resist the turgid pressure of the cell. So the turgid pressure is resisted by the cell wall that we have got it in the term of A, and we would say this is cell wall. They say what controls activities of the cell. So we know that that is the nucleus, and this was C with us. So the letter that we have here is C, and we have got the name of the cell structure, which is the nucleus. And at the end, they ask us that what is the site of the chemical reaction of the cell, including the synthesis of the protein. So here he has not just asked the making of the protein, but he is saying that what is the site of the chemical reactions, including the synthesis of the protein. So we know this is the cytoplasm, and the letter that we have to be writing down here. So this is B with us. Moving onward, if we go on toward the B part, they say that the same biologist, what he actually did, he took few drops of the concentrated salt solution and he immersed the cells, those epidermal cells that he took from the section of the plant, and then he started to look under the slide at what appearance these cells would have, owing to the fact where he added these few drops of the concentrated salt solution. So if I were to zoom in, what we see as of now is that the cell membranes, they have pinched away from their respective cell walls. And when that is the situation, what you need to be keeping in your mind is that the plant cells, they are becoming plasmolized, because the issue here is that since the scientist, he added few drops of concentrated salt solution, so this concentrated salt solution that we have got on the outside of the cell, so this has lower water potential in it. So when we have got lower water potential on the outside of the cells, so automatically the water would move out of the large vacuole of these cells and it would go into the surrounding concentrated salt solution. And as a result of that, the cell membrane has pinched away from their respective cell wall, causing this plant cell to be becoming plasmolized. Now, if we were to be going on toward the questions, so here for the first part where the marks are 3, they are asking that you have to provide a reference to figure 4.2, where we are required to be describing the effect on the plant cells where we have added the concentrated salt solution. So since we just need to describe it, so what we would say that the plant cells, they have become plasmolized. The cytoplasm or the large vacuole, you would say that it would decrease in volume, and the cell membrane, this would appear to look being pulled away from the cell wall. Now, if we were to move further, again for 3 marks, what the examiner is asking is that we are required to be using the term water potential, and we need to be explaining the effect. In the above part, we have just described it. So what you would say that the surrounding salt solution, since it is concentrated, so it has a lower water potential than the plant cell, and automatically within the plant cell, the water potential would be more. So that would compel the water to be moving out of these plant cells, and it would go out with the help of osmosis, and that would be down the water potential gradient, and the water would be crossing the partially permeable membranes in the process. So the above was the description where we simply were required to be giving him the references by just looking on toward the cell which they have shown us. And here in the second part, we have presented the examiner with the explanation. Moving onward to question number 6 that we have with us, and that came in May, June of 2015. And they say is that some plants, they can be grown in water using the technique of hydroponics. You may have looked at somewhere on the social media that in some scenario we don't need to be using the soil, but only the water is required, and in the water the roots of the plants, they are immersed, and that solution in which the roots, they are being immersed, so it has got the ions, and the ions concentration is kept at such a concentration that it would ensure the maximum growth of the plant. They say some ions can be absorbed both by diffusion and the active transports. If I were to just simply show you the diagram, so if I were to take some, you would say pot, and in the pot, I would simply add the water, and that water would have a correct concentration of the ions. And for the plant that I am required to be growing, so we would simply would cause its roots to be getting immersed here, and they say that the ions from this solution, it would be absorbed by the plant roots, both by the diffusion and the active transport. Now he has asked you a question for the total of 2 marks, where he wants you to be stating two features of the diffusion that do not apply to the active transport. Actually, the diffusion and the active transport, they go opposite to one another. The diffusion would move the particles from the higher to the lower concentration. Active transport moves it from the lower to the higher concentration. Diffusion would never require the presence of the carrier proteins, but the active transport would require them. The diffusion does not uses energy, so this is the passive process. And the active transport uses energy which is released from the mitochondria by the respiration. So the active transport is an active process. So I have written 4 points. You can write any two. You would write in term of diffusion that this is passive process, and the particles they move down the concentration gradient. If you forget these 2 points, so you would say that for the diffusion, no carrier proteins are required or involved, and the substances they move down the concentration gradient. Moving onward for the second part, they have said and explained how the roots they are adapted to be absorbing the ions. So now since they are asking the adaptation of the roots for absorbing of the ions, so we know it very well that the roots they have got the root hair cells in them, and those root hair cells they would for sure increase the surface area to the volume ratio for the absorption of ions. And at the same time, the roots we know that they continually try to grow into the soil at a greater distance so that it can find new sources of the water. Other than that, you could say that the roots, since they have the root hair cells, so they have got the presence of the carrier protein, and that allows them to be absorbing the ions. Moving onward, we have got some investigation to be cracking. And if I were to simply zoom in more, so here they are asking that we have got a group of students. And what that group of students are doing, that they have taken some small onion bulbs and they try to soak them in solutions where they have changed the concentration of the sodium chloride in them. As of the protocol, they have taken the onion bulbs and they have removed their outer papery leaves, like you may have seen the onion in your daily life. And they have got that papery kind of leaves on them, and that papery kind of the leaves they are removed off these onion bulbs. And afterward, they divided these onion bulbs into total of 6 batches, and each of the batch it would have total of 10 onions on them. In order to carry the investigation, what they actually did or initially did, that the onions they were surface dried with the paper towels and they were weighed, and the mean mass of the onions in each batch was calculated. Like if I were to just simply show you for the first batch, so I have got the first batch of the onions, and in this batch, what I have done that I have placed total of 10 onion bulbs, and these 10 onion bulbs I have cut them, and then we have surface dried them, and then we have taken their initial mean mass. And afterward, these onions they would be added to the sodium chloride solution for the total of 3 hours. And after 3 hours, what they are doing next is that they are removing these onion bulbs out of the sodium chloride solution, and they would again surface dry them and they would weigh them again. And afterward, they have given you their results in the table 2.1. Now, the understanding of this table is very much important. Like, if you were to see, we have got zero concentration of the sodium chloride. Like, they would simply take the water and they would not add any of the sodium chloride into it. But moving onward, per dm cube, they have now added 25 grams, then they added 50 and 100, 150 and 200 gram. And if we just look on toward the mean mass of the onion bulbs in term of the gram, so before soaking, it was 147 after 3 hours. The mass of the onion bulbs has increased from 147 to 173, and the overall percentage change in the mass of the onion bulb, this is indicated as positive 17.7, which is indicating that at this concentration of the sodium chloride, the water has entered into the onions, and that has accounted for an increase in the mass. And the same is happening at a sodium chloride concentration of 25. But if we look on toward 50, 100, 150, so you see that the values that we have been given at then in terms of percentage change in mass, so they are coming in negative. And the final mass of the onion bulbs after soaking, this is less than the initial mass. So this actually is indicating that at these concentration of the sodium chloride, the onion bulbs have lost the water. So that is why we see a decrease in their percentage change in their mass. And we were to simply move on toward the question, and this is a numerical that we are required to be solving. They say calculate the percentage change in the mass of the onion bulbs that were in the most concentrated solution of the sodium chloride, and we are required to be showing the working. And they say that you are required to be writing your answer in the table 2.1. Now, if I were to move back, so since I have to be calculating the percentage change in mass, So the final mass after soaking is 175. The initial is 183. So I would take uh, the difference among these values. Then I would have to be dividing them by their initial mass, and then I have to be multiplying the value with the 100. So to be getting the percentage change in mass. So here you see the final is 175. The initial is 183. I am taking the difference of these two values. Then I am dividing them by their initial mass, and then I am multiplying it with 100. So this comes to be minus 4.4 with us. So now we are writing it in the table. If we were to move onward, they say that explain why the student they calculated the percentage change in mass of the onions, like the percentage change they are calculating. The reason is that the initial mass of all the onion bulbs, this basically is different. So when he would be calculating the percentage change in mass, then for him it would be easy to be comparing the change that we have got in terms of their percentage change. So if I were to just simply take you to the answer for 2 mark, so we would tell our examiner that the starting mass of all the onion it was different. So if we would calculate the percentage change in mass, so this would allow us for a valid comparison, and that would be helpful in determining that what water potential we have got for the onion bulbs. Now, again, we have got this table, the graph with us, and this graph says that the student they plotted a graph of the results, and that is shown in figure 2.1. And again, we are required to be looking on toward the x-axis, which is showing us the concentration of the sodium chloride solution in terms of gram per dm cube, and this is increasing from 0 to 200. And here they have given you the percentage change in mass, and this is coming on toward the y-axis. So this is our dependent variable. Now the idea here is that at all these concentrations of the sodium chloride that goes from 0 till this point, which is 45. Now, why I am indicating this point as 45? Because between 0 to 50, each small box is representative of 5 units. So here we have got 45. So when the sodium chloride concentration is being increased from 0 to 45, you see that we have got the value of the percentage change in mass in terms of a positive value. So this means that at all these provided sodium chloride concentration, the onion bulbs, they are gaining water, so their mass is in the positive value. And if we just simply come at this point of 45 gram per dm cube of the sodium chloride concentration where the graph is touching the x-axis. So at this specific point, the water potential of the onion bulbs and their surrounding sodium chloride solution is same. So we see no overall change in their mass. But as the concentration is being increased from 45 above, so we see a decrease in the percentage change in mass. So this means that from 50 till 150 gram per dm cube of the sodium chloride concentration, the onion bulbs they are losing the water. The water is going out of them into the surrounding sodium chloride solution. So that is why we see a decrease in their percentage mass. Now here the examiner wants us to be completing the graph, and we need to be including our answer to B first part. So if you just look on toward the graph, they have given you the markings till 150 gram per dm cube concentration of the sodium chloride. And if I were to move back toward the table here, so you see that we have got this 200, and against this we have got the value of minus 4.4. So if I just revert back to the graph, so I need to be highlighting here instead where would minus 4.4 would lie. So if we were to look downward, so each small unit is representative of one value. So here is 4 and minus 4.4, it would come somewhere here. So if I just drag it onwards to be putting a value on the graph, so the 4.4 is coming at this point against a value of 200. So I need to be putting a cross here, and then I would have to join this marking to this marking. I hope this would make sense to all of you. And for the second part, they say use the graph in figure 2.1 to estimate the concentration of the sodium chloride solution that has the same water potential as onion. So that concentration of the sodium chloride where the water potential of this solution and the onions would be same. So we would see no overall percentage change in mass, and that would be zero. So I need to see where the curve touches. So this I have already highlighted that this is 45 gram per dm cube concentration of the sodium chloride, where there would be no overall change in the mass of the onion bulbs. So moving onward for the D part, the examiner has given total of 4 marks, and they are asking that the students they are required to use the term water potential, and they are required to explain why the onions they have gained the mass when they were soaked in dilute solution of the sodium chloride. And for the second part, they say that why these onion bulbs they are losing the mass when they were soaked in concentrated solution of the sodium chloride. So if I were to explain it separately, now you have to be basically assuming is that if I were to have a beaker, and if in this beaker, if I would add the dilute sodium chloride solution, and at the same time, we know that the scientists they have immersed the onion bulbs into it. So you see that this dilute solution has higher water potential in it, and the onion bulbs that are immersed in it, their water potential is low. And we know by osmosis, the water molecules they would move from higher water potential that is there in the dilute sodium chloride solution, and they would go into the onion bulbs where the water potential is low, and that is why in dilute sodium chloride solution the onion bulbs they would gain the mass. Now if I were to reverse the situation, where if we make the outside solution not dilute, but rather than if this becomes a concentrated sodium chloride solution, so that would basically mean that now its water potential comparative to the onion bulbs is lower, and the onion bulbs would for sure have a higher water potential, and by this through osmosis the water would exit these onion bulbs into the surrounding sodium chloride solution, and when the water would be lost, so for sure the onion bulbs they would lose the mass. So if I were to simply like read the answers for you for the first part where we have immersed them in the dilute solution of the sodium chloride, so we would say that the dilute solution of the sodium chloride has higher water potential than the onions, and this allows a water molecule to move into the onions where the water potential, as I have said, is low, and that would be done with the help of the osmosis, and by that the onion bulbs they would gain the mass. On the other way around, where they are losing the mass when we soak them in the concentrated solution of the sodium chloride, so we would say that the concentrated solution of the sodium chloride has the lower water potential than the onions, and that would allow the water to move out of the onions, and we know in the onions the water potential was high, and that again has happened with the help of osmosis. And when this would happen, so the onion bulbs they would lose the mass. So by this way you would score total of 4 marks for this. Now if I were to move further, they say for question number 7 that came in the May June of 2016 and the variant was 41, they ask us that the Rhabdostyla is a single celled organism and it has no cell wall and it has got no chlorophyll. Further, they say that these gases they are exchanged across the cell membrane of the rhabdostyla, and they want us to name the gas which is produced by the rhabdostyla. So the answer is very simple that these rhabdostyla they would respire, and when they would respire, so the gas being produced in them is the carbon dioxide gas. And the second point they say is that the process that produces the gas, so we know it is a respiration where they would combine the glucose with the oxygen gas and that would produce the CO2 gas, and this process would be the respiration with us. Now since this is a single celled organism, so it would not have a specialized respiratory system. So when they are asking that we are required to be telling him the method of the removal of the gas, so the answer is very simple that this would be by the diffusion process, because wherever the respiratory gases they are being exchanged between the organism and the surrounding environment, and specifically for a single celled organism, you always have to be stating him that this is the diffusion process. If I were to move further for this figure 4.1 which they have shown you, so the information the examiner is giving that this Rhabdostyla this lives in freshwater habitats such as ponds and the lakes and the rivers that we have got. And this fresh water has got a very low concentration of the solutes, and this is indicating that in the fresh water, the water potential it would be high, and that would mean that the water would be entering into the body of the Rhabdostyla. The problem with them is that since they do not have the cell wall in them, so if the water would keep on entering in their bodies, so this means that their bodies can burst or Rhabdostyla can burst. Now further they tell us that these Rhabdostyla they have a contractile vacuole that fills with the water and empties at an interval as shown in figure 4.1. And further they tell us is that this contractile vacuole this would remove the excess water out of this Rhabdostyla. Now if you were to see the first diagram of this Rhabdostyla, so we have this contractile vacuole, and you know that when the water would be entering into the Rhabdostyla from the surrounding water, because the surrounding fresh water has a very low concentration of the solutes, so you see that they are filling up their vacuole with excess water, and that vacuole this is fusing with their cell surface membrane, and by that they are removing the water out of their body. And this is a strategy that we see by which the Rhabdostyla would get rid of the excess water, and they would not burst owing to the fact that they do not have the cell wall in their structure that can prevent their bursting. Now, if we were to just look on the question, they say for 3 marks, they ask us that you have to explain using the term water potential that why Rhabdostyla needs to remove excess water. So, what you would tell him that the water that is there in the surrounding of the Rhabdostyla it has a higher water potential. So it would enter into the Rhabdostyla from higher water potential in the fresh water to the lower water potential, and that too would move down the water potential gradient by osmosis through the partially permeable membrane of this organism, and as a result of it the water would fill up the Rhabdostyla and ultimately it can burst it. Now, this is something Rhabdostyla cannot afford, so it would remove this excess water with the help of those, you could say, the contractile vacuole that they have got in them. So ultimately, they are removing it. Now, if I were to go further, so they have given you a graph here, and they say that they have carried on an investigation in which they took the individual Rhabdostyla and they were placed into different concentrations of the seawater. Like for sure, there would be a situation where we would have a beaker, and in the beaker, what we are adding into it is the seawater. And as far as the seawater is concerned, so they are changing the concentration of the salt in the seawater, and they are actually trying to see that how come the Rhabdostyla would be excreting the water out of them. So they say the rate of the water excreted by the contractile vacuole of each organism was determined, and they are showing you the results in figure 4.2. Now for the graphs, the situation is that we need to be looking on toward the labelings of the y-axis and on toward the x-axis. The x-axis gives you the concentration of the seawater, and the y-axis is giving us the rate of the water excreted, which is in term of micrometer cube per second. And if we could simply see that when the concentration of the seawater is increasing, the rate of the water excreted by their contractile vacuole, this is decreasing. So what this implies is that when I am increasing the concentration of the seawater, so the salt in them would be increasing and the water would be decreasing. So as the concentration increases, less water would be entering into the Rhabdostyla species. And that is why they would not have the urge to be removing the water which is entering into them. For sure, when the concentration of the sea water is zero, so a lot of water would be entering, or the excess water would be entering into the Rhabdostyla. So that is why they are removing more of it by their contractile vacuole. At 4 concentration of the sea water in term of percentage, less water is entering into the Rhabdostyla comparative to this concentration. So for sure they are removing the water, but the rate of the water removal is less. And at 12 concentration, very minimum water would be entering into the Rhabdostyla. So they would hold it rather than to be removing more of it by their contractile vacuole. So now they ask us for a total of 3 marks that we are required to be explaining the results that are shown in figure 4.2. So you would simply say that as the concentration of the seawater increases, the removal of the water out of the rhabdostyla, this decreases. Like if you could simply see that as the concentration is increasing, less of the water is being excreted out of these rhabdostyla, and for sure this would decrease the steepness of the water potential gradient that would exist between the rhabdostyla and the surrounding water. So for sure, less water would enter into them when the high concentration of the seawater is available, and that would require the Reptostyla to be removing less water out of their bodies. Moving onward, for the last part, they say that single-celled organisms with cell walls do not have a contractile vacuole in them, and we are required to be giving him the suggestion. Now, the idea is that the organisms, those who have the cell walls in them, so these cells wall, they are inelastic and they are rigid. So when they would fill up with the water, so their vacuole or their cytoplasm, it would exert the pressure on the cell wall, but since the cell wall is inelastic and the rigid, so ultimately this would prevent the entry of the excess water and they cannot burst. So they do not need to have the contractile vacuole to be removing the excess water rather than their cell wall would be doing the purpose. So how we are required to be answering it, that the cell walls of the single-celled organisms are inelastic and rigid. So they would resist the turgor pressure which is exerted on their cell walls by the water. And when the cell wall is, you could say, resisting the pressure, so this would resist the extra entry of the water, and the volume of their cells would not change, and this would be preventing their bursting. So this is how you would score total of 3 marks for this. Now, if I were to move further, again they ask a question from the osmosis. This appeared in May, June 2016, and the variant of your paper was 4.2, and they're asking you to be defining the term osmosis, and we have done it in a lot of question above, that the osmosis is the net movement of the water molecule, where it moves from that area where we have higher number of the water molecule, and that is the area of the higher water potential, and they would move to an area of lower water potential where the number of the water molecules is less, and this would be done down the water potential gradient. And for sure, the water, when this moves by the osmosis, so this for sure needs to be crossing the cell membranes, and the process would be depending on the internal kinetic energy of the water molecule, and no energy would be utilized from the mitochondria. So that is why we are adding the word at the end, and the word is passively, that for osmosis no energy is utilized. Now, if we were to move on toward the part B, so in the part B, they say that we have got these students and they measured the mass of 4 of the potato sticks using an electronic balance. So they would have some kind of the potato sticks and they have used this electronic balance, and by that they are basically measuring their mass. Now at the moment we don't see any potato stick being present in this dish. So the value of this apparatus this goes as 0.00 gram, like if you were to look clearly. Now moving onward, what they say is that these students they left each potato stick in one of the 4 different liquids for the total of 5 hours. Now, if you were to look here, we have got distilled water, we have got 0.1 mole per dm cube sodium chloride solution, then we have 0.5 mole per dm cube, and at the last we have got 1.0 mole per dm cube sodium chloride solution. So for sure there would be total of 4 beakers, and in all of the beakers they are putting one of the potato sticks into each of them, and after immersing them for the total of the 5 hours, now they are again measuring their final mass again, and they are calculating that what change in the mass has occurred. So what you need to keep it in your mind is that in the distilled water, if we would see an increase in the mass of the potatoes, so that means that the water has entered into the potato stick. And any of the concentration which they have given us for the sodium chloride solution, we have to see it that whether we see an increase in the mass or a decrease in mass. So never you have to consider is that if you come across the word sodium chloride solution and they have given you and their concentration as 0.1 mole per dm cube in this situation, so this never suggests that the water is exiting the potato sticks, it can also enter, because the student they usually believe that wherever there is presence of the salt, so the water would always leave the potato sticks or the potato slender, but this is not always the case. Let us move onward, and here they are asking us that we are required to be predicting that which of the liquid would cause the largest decrease in the mass of the potato sticks. So for sure we need to be highlighting or we need to be mentioning the sodium chloride solution, which is the most concentrated, and this is 1 mole per dm cube. Sodium chloride solution where the water potential is the least. So more amount of the water would escape out of the potato sticks into this solution of the sodium chloride solution, and that would account for the maximum loss in the mass. So the answer is very simple. We would say this is 1 mole per dm cube sodium chloride solution. Now, if we were to move further, they say the student they dried the potato sticks with the help of the paper towel before putting them on the electronic balance, and they are asking a suggestion that why they have done it. So when you would cut the potato sticks out of the genuine potato or the original potato, so some amount of the water is always available on their surface. So we need to be removing this excess water that is present on their surface. So after when we immerse them in the distilled water or the sodium chloride solution, then when we can get an accurate measure of how much there has been an increase or a decrease in the mass of the potato sticks. Now, moving onward, they say that after the experiment, the student noticed that the potato sticks with the lowest mass was soft and floppy. And now, when they are saying that the potato sticks have become soft and floppy and they have got the lowest mass, so this is very evident that the potato sticks they have lost the water. The water has gone out of them into the surrounding solution, and as a result of that, their large vacuole it would decrease in size, and their cytoplasm would also decrease in volume. And as a result of that, the cell membrane they would push away from the cell wall, and none of the cytoplasm of the large vacuole is now exerting pressure on their cell wall, so they don't appear turgid, but rather than they become soft and floppy. So here they have asked for the total of 3 marks that we are required to explain why the potato sticks have become soft and floppy. And the answer is very simple that the potato sticks they have lost the water by osmosis, where the water has moved from the higher water potential, which was there in the potato sticks, and into the surrounding solution where the water potential was low. And in this way, their large vacuole decreases in size or the volume, and by that, the large vacuole is no longer exerting the pressure on the surrounding cell wall, and that is why they would become flaccid or they would become soft and floppy. If we were to move further for the total of 2 marks, they are asking that the student followed the same experimental procedure, but now they did not take the fresh potato sticks, but rather than they boiled the potato sticks and now they are immersing them into the solutions. And what they measure is that there is no overall change in the mass in any of the solution, like whether they place them in distilled water or whether they place them in the other solution that was 0.1, 0.5, and 1 mole per dm cube. So the boiled potato sticks, they do not show any change in the mass. So this means that once we boil the potato sticks, so no osmosis is occurring. Or other than that, we could say is that there is no net movement of the water molecule in case where they are boiled. So the answer becomes simple that when you would boil them up, so their cell membranes are damaged. And when their cell membranes are damaged, so ultimately no osmosis occur. Maybe the situation would be that the amount of water that would be leaving the boiled potato sticks, it would be equivalent to the water that is entering into their potato sticks, because we know in osmosis the cell membrane is always involved, and by boiling when we denature or when we damage the cell membrane, so automatically osmosis would not occur, and as a result we would not see any increase or the decrease in the mass of the potato sticks. Now let us come to this question number 9 that we have got, and this appeared in your May June of 2016, and the variant was 4.3. They say we have got the student and they investigated the diffusion of the substances. Now here we have to be focusing is that they are determining the diffusion of the substances, and that is actually being done through a whisking tubing. And in the biology experiments, the whisking tubing basically acts as an artificial membrane which has the properties as of the cell membrane, and that we have to perceive is that these whisking tubes they would be acting as a partially permeable membrane where they can control the entry and the exit of the substances. Now, what they have to tell us is that the student they made a bag of the whisking tube and they are showing you in figure 4.1. Now, if you were to look this diagram, so you see that we have got a top of the plastic syringe, and at the end of the plastic syringe, they have attached the whisking bag. They have placed a knot at their end, and then they actually tighten it up with the help of this elastic band. Now, if you were to see further, what the students here are doing is that in this whisking bag, they are filling it up with the help of the starch solution, and they place this, you could say, whisking bag that is mounted to the top of the syringe. Now they are taking it into the test tube of the water. So if we just see, you have to basically assume here is that this whisking tube, this has got the starch solution, and now we have got the surrounding test tube, and this surrounding test tube, this has got the water in it. But as of the work statement, we need to be binding to this concept of the diffusion of the substances. But if we were just to move further, they say the student they added some iodine solution to the water in the test tube. So this water that we have got in them, they are adding the iodine solution. And we know that the iodine solution, whenever this would come in contact with the starch, so it would turn the color to black. Now, what they have to tell us further is that after 30 minutes, when they kept this at a room temperature, so the content of the whisking bags, they were stained blue-black, but the water outside remained yellow color. So you see that they are not asking us with respect to the osmosis, but rather than we have to speak in terms of the diffusion of the iodine or the starch solution. Now, considering the fact that the iodine solution this was available in the water which is surrounding the whisking bag, so here the concentration of the iodine solution is higher, and within the whisking bag we do not have any iodine molecule as of now, so here the concentration of the iodine is lower. So as a result of that, the iodine molecule they would diffuse from the surrounding water into the whisking tubing, and at the same time the size of the iodine molecule this was small enough that it allowed them to diffuse on the inside of the whisking bag, and where they would come in contact with the starch and it would change their color, and the iodine would change its color to the blue black. But why the surrounding, you could say, water, it remained yellow? The reason is because starch, we know this is a complex sugar. This is a polysaccharide. So starch is large enough that it would not be able to cross the membrane of the whisking bag, and ultimately it cannot diffuse or it cannot move into the surrounding water. So that is why these contents on the outside of the whisking bag, they do not change color, but rather than here, since iodine has moved on the inside, so now it will turn blue black. So now they have asked for the total of 4 marks that we are required to be explaining the result as I have explained. So now you just need to know the wording. We would say that the iodine molecules are small, so that is why they would move from the water in the surrounding test tube into the whisking bag, and that would be done with the help of the diffusion. And that is why they would stain the starch and its color would turn blue black. But if we talk on the other way around, the starch which was available in the whisking bag, so this was large, and that is why it cannot diffuse or it cannot pass out of the whisking tubing into the surrounding test tube, and that is why the color of the water on the outside this would remain yellow color. If we were to move toward the second part for the total of 3 marks, they are asking that we are required to state three factors that influences the movement of the molecules through the membranes. So you would say that this is a temperature because more temperature there is, so more movement of the molecules there would be through the membranes. The other thing is the surface area. We have the greater surface area, so it would allow for the greater movement of the molecules. And the third could be the size or the type of the molecules. Because if the size of the molecule is enormous, so ultimately they cannot move through the membrane. But if their size is small, so they can move through the membranes. The other alternative point that you could write instead of the size, so this is the type of the molecule. And why the type of the molecule is important? The reason is that some of the molecules, like the charged particle as we have the ions, so they cannot pass through the membranes, rather than they require some channel or the carrier proteins. But if we have got some non-polar or non-charged particle, then ultimately they are in the position to be crossing these cell membranes that we have with us. Moving onward to this question number 10. This is from the active transport, and this came in the May June of 2017, and the variant of your paper was 4.2. And they asked that figure 2.1 that we show you. This is a diagram of the protein, and this protein is used to move the ions across a membrane, and there too in the root hair cells. Now, if I were to explain you this diagram, so here we have got this inside of the cells where there is higher concentration of the ions. And this darkened part is showing you the cell membrane. And here we have got the outside of the cell here where the concentration of the ions is lower. And if you were to follow this arrow, so the ions they are moving from their lower concentration to the higher concentration. So the point that must click in your mind is that since the particles they are moving from the lower to the higher concentration, so this is the active transport. And they have shown you the presence of the protein that is accommodating the movement of the ions. The reason is that whenever the ions have to be moving, so they cannot do so directly through the cell membrane, but rather than there is need or there is requirement of the protein, and these proteins they are the carrier proteins. So they facilitate the movement of the ions because at time the ions make a contact with these proteins that we have in the cell membrane. So these protein or the carrier protein they would change the shape, and that would allow the ions to move on the inside of the cells. Now they ask us for 1 mark that we are required to state the name of the process that moves mineral lines into the root hair cells through the cell surface membrane. So the process is active transport. Now for the total of 3 marks, what they have to be asking is that we are required to explain that how the protein molecules they move the ions across a membrane during this process. So since active transport is being utilized and the protein molecules are being utilized, so we need the energy being released from the mitochondria by respiration. So we would say that the protein molecules they use energy that is released by the respiration. And by that the ions they would move from the lower concentration which was on the outside to the higher concentration which was on the inside. And that too is happening across a membrane. So this all elaboration is for the active transport. And if we have to be giving weightage to the protein word which the examiner has used in the question, so we would say that these proteins they would change the shape when they interact with the ions. So that would facilitate the passage or the movement of the ions on the inside of the cells, and by this you would score total of 3 marks for this. If I were to go beyond, so this is question number 11. This came in the year 2020 for October, November, and the variant was 4.1. And they say water is an essential molecule of life. We are required to be completing the statement, and the marks are 2. Now they are talking with respect to the movement of the water, and they say water moves into and out of cells, and we know this is always with the help of osmosis. Further, they say is that the water is known as this because it can dissolve solutes. Now we know that anything that can dissolve the solutes in it, so we would always have to be referring to water as a solvent. If we were to move to the part B, which they have shown us, they say that we have got a leaf cell and this was put into a solution. Like we have placed the sleeve cell into a solution, and the water potential of the solution was lower than the water potential of the content of the cell. Now you see that if I were to talk about the solution which was here on the outside of the plant cell and it has a lower water potential, so that would mean that the cellular content they would have a higher water potential and the water would move out of the cells, and as a result of that the large vacuole and the cytoplasm they are decreasing in volume, and the cell membrane they appear to look pinch away from the cell wall. And this is why this plant cell as of now looks flaccid or this looks floppy. Now what they have to tell us further is that the same leaf cell this was transferred into the pure water. And now they want us to sketch the expected appearance of the cell after it had been in the pure water for 3 hours. Now, if I just replace the surrounding solution with the pure water, so that means that on the outside the water potential would be high, and in the large vacuole or the cytoplasm of the cell, the water potential would be less. So by osmosis, the water would enter into the plant cell. And that is why the cytoplasm would increase in volume, the large vacuole would increase in volume, and the large vacuole would exert the pressure on the cell wall. So these cell walls in the sketch, we need to be showing it a little bulged out. So that means that the examiner would be convinced that the student knows that here the plant cell is becoming turgid. We are required to be increasing the size of the large vacuole in the sketch, and we would move the nucleus to little bit of the corners, and they would also be pushed these chloroplasts which they have given us. So if I were to just simply take you to the diagram for 3 marks, so you see that I have bulged the cell walls out, I have increased the volume of the large vacuole, I have increased the size or the volume of the cytoplasm as well, and I have made sure that the cell membrane, they are now in contact with their surrounding cell wall. So if you would make a diagram like this, so you would score total of 3 marks. If I were to move further, they say that we have got a plant and that plant was not watered for 1 week. And they say that this figure 1.2 that we are showing, it is a series of the photograph of the plant during the week. And you see that here on the day 0, the plant looks upright. And as the day are passing to day 3, the plant is wilting, isn't it? Now, what you are required to be understanding here is that if I would not water the plants for 1 week, so this means that the water would not enter into the cells of these plants, and when it would not enter into the cells of these plants, so this means that the large vacuole would not fill up with the water. And if this would not fill up with the water, then as a result of that, the large vacuole would not exert the pressure on the cell wall, and these plant cells they would lose their turgidity. And since a lot of leaves are being shown, so this means that here we have got the palisade cells. So when the palisade cells are no longer turgid, so they would not be able to support the upright position of this plant. Now, if we were to go to the question for the total of 3 marks, what the examiner is asking, that we have to explain how the lack of water has affected the support of the leaves of the plant, shown in figure 1.2, and we are required to use the term turgid pressure in our answer. So the answer is very simple. I have explained it above. You just need to look on toward the wording that the students they were required to write down. You would say that since the water was not provided to these plants, so it would not enter into the large vacuole. And when this is not entering into the large vacuole, so the large vacuole would not exert the turgid pressure on the cells wall of the mesophyll cells, and ultimately they would collapse. And by the end of the week, the plants they would appear wilt. Moving onward to this question number 12, this is coming from the diffusion question. And this was variant 4.1 for your year 2021, and it was session October-November. They say figure 1.1 is a micrograph of the part of some fish gills. So we see a lot of fish gills here, and if you were to see, they have got a lot of tiny projections. So this means that here the surface area of these content is greater, and they would also be very thin because this is a very higher magnification which they have shown you. So they are asking is that the fish gills they are adapted for the gas exchange by diffusion. And for the total of 2 marks, they want us to be defining the term diffusion. And we know in diffusion, the particles they would move from an area where they are in more number or they are in their higher concentration to that area where the particles are in lower number or in lower concentration. And that basically is done down the concentration gradient, and we are not in the need of any of the energy. So that is why we can also write the word passively here. Now, for 1 mark, they have asked that you are required to be suggesting one adaptation which is visible in figure 1.1 that would show that the fish gills are efficient structures for gas exchange by diffusion. So either you could say that they have the large surface area, or you could say that these fish gills they are thin. So that would allow for the efficient exchange of gases by diffusion for these fish gills which they have shown us. Moving onward to this question number 13 that we have got. So this appeared in paper variant 4.1, and this came in May June of 2022. What they have to be telling us is that the chloride ions they also move along the pancreatic duct. Now where the pancreatic duct is lying, we know that we have got this liver, and this liver acts as an accessory organs for our alimentary canal. And moving onward here, we have got the gallbladder. If I were to move further, so we have got this pancreas. And from the pancreas, there is presence of this duct that we have here. This is the pancreatic duct. And this pancreatic duct, this carries the digestive juices from the pancreas to the first part of the small intestine, which is duodenum. Now, what they have to tell us further is that there is presence of the proteins, and these are the CFTR proteins, and they would be available in the cells that are lining the pancreatic duct, and they move the chloride ions out of the cells into the duct. If I were to just see and to move you here, so we have got some cells here in the pancreatic duct. Okay, and these cells in the pancreatic duct, they have got some proteins in them, and they are saying that these proteins they are entitled as CFTR proteins. And these CFTR proteins, they are actually moving the chloride ions out of them, and these chloride ions they are getting accumulated in the lumen of this pancreatic duct. So what you have to get an idea here is that when there would be the presence of the chloride ion here in the duct, so this chloride ion would lower the water potential in them. And for the cells, the water potential would increase, and the water would move out of these cells with the help of osmosis, and it would come here. And that would make the pancreatic secretions or the pancreatic juice so that they could flow easily to the duodenum of the small intestine. Now, if we were to look on toward figure 3.2, which they have given us here, they say that this is a diagram of the cell that is lying in the lining of the pancreatic duct, showing the location and the activity of the CFTR protein. So, if you were to look clearly, this is the pancreatic duct cell. These are their carrier protein, and which are the CFTR protein. And these CFTR protein, they are moving these chloride ions out of them into the duct of the pancreas. So they say for the total of 3 marks that we have to explain that how the CFTR protein they move the chloride ions across the membrane of the cells which are shown in 3.2. So if I were to simply see on the outside of these pancreatic cells, the concentration of the chloride ion is higher, and on the inside, the concentration of the chloride ion is lower, and these ions are also moving with the help of the carrier protein. So all of these, they are the hints that we need to be talking with context to the active transport. So if I were to just simply take you to the answer, so I would say that these CFTR proteins, they would move the chloride ion against a concentration gradient. Why? Because they are being moved from the lower to the higher, and that is against the concentration gradient. And for sure, that would be done with the help of energy, which would be released by the mitochondria. And we know mitochondria, they basically release energy with the help of aerobic respiration. And these carrier proteins that we have got, now they would interact, or they when they make the contact with the ions, they change their shape, and that would facilitate the passage of the ions out of the pancreatic cells, and ultimately they are moving into the pancreatic duct. Moving further, for the total of 3 marks, they say is that the movement of the chloride ions into the pancreatic duct causes the water to move from the cells into the duct, and that would help the flow of liquids in the duct. They say explain how the water moves from the cells shown in figure 3.2 into the pancreatic duct, and as I have explained above, you see that we have got a lot of chloride ions here that would reduce the water potential on the outside of these pancreatic duct cells, and inside the cells the water potential it would for sure increase, and that implies that now the water would move out of the pancreatic cells into the pancreatic duct with the help of the osmosis, because the presence of the chloride ions here, their higher concentration, it actually reduces the water potential. So simply if I just take you to the answer, for 3 marks you would say that the presence of the chloride ions in the duct, it lowers the water potential, and it causes the water to move out of them by osmosis, where they would move from an area of higher water potential, which was lying within the pancreatic cells, to the lower water potential, which was there in the pancreatic duct, and that has been done across a partially permeable membrane. So by this, you would score total of 3 marks here. Moving onward to question number 14 that came in May June of 2023. The variant was 4.1 and the question number was 2. And here we have got the students, and these students they started to investigate the osmosis that too in the potato plant cells. Now what he actually did, that out of the potato he has cut their cubes and then he immersed them in the water, and that water had got different concentration of the sucrose solution, and then he kept these cubes of the potato tissues for the total of 30 minutes into the water that has got different concentration of the sucrose solution in them. Now, what they actually did that before immersing the potato cubes into the sucrose solution, they would have measured their initial mass, and after putting them into the 30 minutes, now they would have measured their final mass, and now they are calculating the percentage change in the mass, and the results are shown in table 2.1. Now, if I were to explain you the table very clearly, so in the first column, they have shown you the concentration of the sucrose. Then this was the mass of the potato cubes before immersion in terms of gram. This was their mass after immersion for 30 minutes. And here they have calculated the percentage change in mass. Now you were to see that as we move down the column, the concentration of the sucrose solution is increasing. And if we were to see the first three concentration that ranges from 0.00 to 0.40, so you see that the final mass is greater than the initial mass. So this means that at these concentration of the sucrose solution, the surrounding solution has higher water potential, and the potato cubes they have less water potential, and the water entered into them, and that is why their mass is increasing. And we see the value for the percentage change in mass as being in positive. But if we were to look the last three concentrations ranging from 0.60 to 1.0, so the final mass of the potato cubes, this is less than their initial mass, and that would indicate that these would be the concentrated solution where the water potential was less than the potato cubes. So the water, it left the potato cubes, it came into the surrounding solution, and that is why they are losing the mass, and the percentage change in their mass is indicated with the negative sign. This negative sign would always indicate that some tissue, as in this case potato tissue, this has lost the water, and that has resulted for a decrease in mass. Now they say using the information in table 2.1, we are required to calculate the percentage change in mass at 1 mole per dm cube. So you see we are given the value here, but this percentage change in mass is not available. So how the formula would go? This is the final, this is the initial. We would subtract the initial from the final, and we would divide the value by the initial, and we are required to be multiplying it with 100 to basically see the percentage change in mass. So if I were to take you to the answer, so the final mass is 1.11 and the initial mass is 1.28, and we are taking their difference and we are dividing them by their initial value of 1.28, and then we are required to be multiplying it with 100, and the answer for us, since we have to give it up to two decimal places, so this is 13.28 that you are required to write it here. If now for the second part, we are required to use information which is given in table 2.1, and we are required to explain the difference for the results that they have given us for two concentrations. One is 0.6 mole per dm cube sucrose solution concentration, and the other one is 0.8 mole per dm cube sucrose solution. And the examiner is clearly hinting that you need to be writing the term water potential in your answer, and the total marks given are 5 here. So if I just take you back toward the readings that we have got for 0.60 mole per dm cube and 0.80 mole per dm cube. So what I see is that for this higher concentration of the sucrose solution, the percentage change in mass is far more greater for the percentage which they have given us for 0.6 mole per dm cube. Now, what you have to be basically understanding here is that if I were to have a beaker here, and if the surrounding concentration was 0.6 and that too in term of mole per dm cube, and if I were to have a second beaker, and here if I would keep the concentration of 0.8 mole per dm cube, and if I would immerse the potato cubes into both of these, so the idea is simple, that at both the concentrations, since we are seeing a loss in the mass of the potato cubes, or we are seeing a percentage change or a decrease in their percentage change mass. So this simply implies that the water has moved out of the potato cubes into the surrounding solution, because in the surrounding solution the water potential is low, and in the cubes, the water potential is high. So the water would move out of the cubes into the surrounding solution, and that is the reason we see a percentage decrease in the mass. But the other fact is that there is more loss of the water at 0.8 mole per dm cube comparative to this, because at this concentration the water potential in the outside solution is far more less as compared to the water potential that we have here. And since we have far more less water molecules in this solution comparative to this, so there exists more steep gradient for the water potential. So ultimately, this is losing more water comparative to this. So this is an overall idea that we have to present in our answer for 5 marks. So how you are required to be writing the answer to such questions? You would say that 0.8 mole per dm cube solution has water potential which is far more less than 0.6 mole per dm cube. And for 0.8 mole per dm cube solution, the steepness of the water potential gradient is more, and that would cause its potato cubes to cause its water molecule to move out of the potato cubes from higher water to lower water potential. And you see, we are actually complying with what the examiner has asked us. And that is why the loss of the water out of the potato cubes or the cylinder is relatively more in this higher concentrated or 0.8 mole per dm cube solution than 0.6 mole per dm cube solution. And that is why the potato cubes, they are losing more of the mass at 0.8 mole per dm cube than 0.6 mole per dm cube. Now, moving onward for the total of 2 marks, they say that we are required to describe the expected appearance of the cell from the potato cube that has been immersed in the distilled water for the total of 30 minutes. So you see that if I would place the potato cubes in distilled water, so the water would enter into their large vacuole. So their large vacuole or the cell membrane, they would increase in size, and for sure they would put the pressure on the cell wall, and the plant cells, they would become turgid or the swollen. Like we were required to be writing the appearance, not the mechanism. Now, for 3 marks, they're asking that we have to describe how the process of active transport this differs from the process of osmosis. So we know it very well that the active transport, it uses energy which is released from the respiration, and the active transport would move the particles against the concentration gradient, and the active transport would cause the movement of the ions with the help of the carrier proteins, and none of these situations we see in case of osmosis. Moving onward to the C part, they say state the type of the plant cell that uses active transport to absorb the mineral ions from the environment. So this is fact that these are the root hair cells, because the root hair cells, they have the abundant mitochondria and they have the carrier protein. The mitochondria would release energy and the carrier proteins would allow the movement of the ions. So the answer here is root hair cell. And for the 2 marks, they ask us that we are required to explain the effect of the lack of magnesium ions on the color of the plant leaves. Now, what we need to be understanding here is that these magnesium ions, they are important for making the chloroplasts or the chlorophylls within the leaves, and those that chlorophyll pigment is green and that would make the leaves look green. So if there is deficiency or lack of magnesium, so the leaves, they would turn yellow. And why they are turning yellow? Because there was lack of the magnesium ions, and these plants, they were not able to make the chloroplast or the chlorophyll. So that is why the plant leaves, they appear yellow. Moving onward to this part 15, they say that the movement of the molecules within the organism can occur both by diffusion and the active transport. And we are required to complete table 1.1, where we have to put a tick to show that what feature is shown by the diffusion or the active transport. So we have got the feature here, we have active transport here, we have diffusion here. And they say movement of the particle always occurs across a membrane. So always word basically implies that we need to be putting a tick here, because the diffusion can happen across a membrane, but the presence of the membrane is not an obligation for the diffusion process. They say movement of the molecules during gaseous exchange. So the gaseous exchange always is by diffusion. And they say rate of the movement of the particle is higher when the concentration gradient is larger. We know that the active transport moves the molecules against the concentration gradient. So the steepness of the gradient would account for the diffusion process. So we need to put a tick here. They say requires energy from respiration, and that would need us to be putting a tick here because this is the active transport. Why it requires energy from the respiration? Because this mechanism moves the ions against the concentration gradient. So for that purpose, we need the energy for the carrier proteins to work so that they can move the ions from the lower to the higher concentration. Now, here they ask for the total of 3 marks where we need to explain that why the active transport is important in the root hair cells. So if I were to explain this with the help of the diagram, so what we know is that the root hair cells, they have got two of the important functions to be rendering. One is the movement of the ions, and the other thing is the movement of the water molecules. And at the center of these root hair cells, what we have got is the large vacuole. Now, the situation is that the mitochondria that we have within the root hair cells, they would release energy, and by that, the ions that we have got, they would move from the soil where they are in lower concentration to the large vacuole, where the ions are in higher concentration. And when we would fill up the large vacuole with more of the ions, so this would lower the water potential within the large vacuole. And now, once we have lowered the water potential in the large vacuole, the water molecules, they would be more in the surrounding soil. That would increase the water potential here, and by that, the water would enter into the large vacuole with the help of osmosis. So if I just take you to the wording that you were required to be writing for this, so you would say that this active transport is important for the movement of ions from the lower concentration that lies in the soil to the higher concentration that lies within the root hair cells. And when the ions are moved through the carrier protein, so they would lower the water potential in the large vacuole, and that would cause the water to enter into them with the help of osmosis process. So by this, you would score total of 3 marks. Moving onward to this question number 16, and this came in year 2024. It was May, June, and the variant was 4-3. And he is asking for the total of 3 marks that we are required to explain that how the water it moves into and out of the animal cell. So the answer is very simple, that the water would move in and out of the animal cell with the help of the osmosis, and the water would always move from an area of higher water potential where the water molecules are more to an area of lower water potential where the water molecules are less, and that too would happen across a partially permeable membrane. If the marks were 4, so you could have added up that this is an entirely a passive process. If we move on toward the second part, what the investigators they have done that they have taken from the person a sample of the red blood cells, and these red blood cells they were immersed into the test tubes that contained the blood plasma, the pure water, and a very concentrated salt solution. And below they have given you the appearance of the red blood cells, and the examiner would like you to highlight that where would the appearance of the red blood cells would be in case of these three of the solutions that we have got. Now, what we know is that the blood plasma has the same water potential as the red blood cells, so the red blood cells would not lose their shape, they would not burst, neither they gonna shrink, so we would write blood plasma right in front of it. If you were to see these cells, you see that they appear shrink. So this means that these red blood cells they were placed in a very concentrated salt solution, and the red blood cells have lost the water as the water would move out of them into the surrounding solution. So this is a very concentrated solution, and the cells they appear like this. And here you see the red blood cells they have burst. So this means that here they were placed in the pure water. The water moved from the surrounding solution from the higher water potential to the lower water potential that would exist within these red blood cells, and without the cell wall, these red blood cells they are bursting. So the solution that you were required to be writing here was pure water. If I were to move further, they ask us that state the name of the molecule that lies in the red blood cells and that would combine with the oxygen gas. So you would always say this is hemoglobin. And for the last question for 1 mark, they say that figure 1.3 it shows the drying of the white blood cells, and we are required to be identifying the type of the white blood cell. Among the white blood cells, we have the phagocytes and we have the lymphocytes. But if we just try to be looking on toward the appearance of the nucleus that is given for this cell, you see this is a multiple lobed nucleus, as there are a lot of lobes of this nucleus. So this is a neutrophil. And the neutrophils, they are a type of the white blood cell that we refer to them as the phagocytes because they eat up the pathogens. Let's move onward to the last question, question number 17, and this appeared in May June 2025. The variant was 4.2. And the figure 5.1 that we see here, they show you the diagram of the root hair cell. So if I were to just simply demonstrate that what lies within the diagram, here we have the cell wall and we have got the cell membranes, and here lies a cytoplasm of the root hair cell. Here comes its large vacuole. And if we were to follow the key, the cross indicates nitrate ions, and the dots that we have got, this is indicating the water molecules. So if you were to simply see, we have got higher concentration of the nitrate ions in its cytoplasm and lower concentration of the nitrate ions in the soil that is surrounding these cells. So if the ions were to move into the cytoplasm of the large vacuole, so what these root hair cells would do, that they would cause their mitochondria to release energy, and that energy would be utilized to move these nitrate ions from their lower concentration that lies in the soil to their higher concentration which lies within their large vacuole with the help of the active transport. And in doing so, these nitrate ions, they are being moved against their concentration gradient, and that too with the help of the proteins that we have got in their cell membranes, and they are the carrier proteins. So this is something they are asking for the total of 4 marks, that we need to use the information in figure 5.1, and we have to explain that how the nitrate ions, they are transported into the root hair cells. So we would say that the nitrate ions, they are moved into the root hair cells with the help of the active transport, where the ions, they are moved from the lower concentration in the soil to the higher concentration, not in the large vacuole, but rather than the examiner, they have showed you the cytoplasm. So we would say that they would move into the cytoplasm against a concentration gradient, and this process would require the energy which is released by the mitochondria through aerobic respiration, and these ions, they would pass via the carrier proteins. So this way you would score a total of 4 marks. And if we were to move further, they say we have to state two ways by which the nitrogen fixation occurs. And by the nitrogen fixation, what we mean is that the atmospheric nitrogen gas, this is being converted into some ions or some form of the oxides of the nitrogen that can be used by the plants. So the two of the mechanisms involve the lightning, and the other one we have got are the nitrogen fixing bacteria that lives within the nodules, within the root nodules of the leguminous plants. For the last part, they say state the name of the process that moves water molecules into the root hair cells. So this goes with the name osmosis. So in this video, I have covered all the questions from diffusion, osmosis, and active transport starting from 2011 and ending in 2025. So I hope you would be very much clear about that in what dimension the examiner gives you the questions and how you are required to be cracking them. So see you for the next class, where I would be talking or discussing the topical past paper from the fourth chapter, which is entitled as the biological molecules.